Hello everyone, welcome to Nuclei's online classes. You are here with me Rasika for Simplifying Biology. In this video, we are going to see about the female reproductive parts of the flower, the pistil, the Megasporangium and the Megasporogenesis. Right, very interesting video this is going to be. So let's start. Yeah, so I hope you remember. Remembering the gist of the chapter is always necessary. That will ensure the continuity of the learning, right? So we saw introduction, we saw about the pollen grain, microsporangium and microsporogenesis. We saw the diagrams of the textbook, and also we discussed some questions as well. So in this video we will see about the female part. Yeah. Okay, so starting with the morphology of gynoecium. Remember what were the members of gynoecium called? The members of gynoecium were called as carpel, right? Carpel or pistil. Carpel or pistil both means the same, right? Now, sometimes this gynoecium will consist of a single pistil, right? Single pistil. If it contains a single pistil, then it is called as monocarpellary, right? Monocarpellary. Mono means single, carpel means the carpel, which is pistil, right? Then you have, sometimes you will have many pistils, right? If you have many pistil, then it is called as multicarpellary, right? Multi means more than one and carpellary means the carpel, right? So that is multicarpellary. And sometimes if you have many pistils, sometimes the pistil will be free like this, right? Sometimes the pistil will be fused. That is, you will have many pistil, but then it will be fused like this. So if it is having free pistils, then it is called as syncarpus. Free pistils means it is called as apocarpus, right? And if it has fused pistils, then it is called as syncarpus, right? See, syngamy means fertilization, fusion, fusing, fused carpels, right? So syncarpus means fused petals, fused pistils, right? Now let's look at the structure of a pistil. So if you look at the structure of a pistil, you will have a receptive platform like this, which is called as stigma. You will have a long stalk like this, and then you will have a bulged end, which is called as your ovary. So that's it. It is your stigma, right? Stigma, your style, and finally the ovary. Right? Now stigma, if you see, it is the landing platform for pollen grains, which means pollen grains will come and attach themselves here, right? So these are your pollen grains. So stigma is nothing but it is the landing platform for pollen grains. It is the landing platform for pollen grains. And what can you tell about the style? Style, nothing. It's just a filament or it's just a stalk, right? It's an elongated stalk, right? Then finally about the ovary, right? So that's the ovary. That bulged part is your ovary. That space that you have inside the ovary is called as the ovarian locule. What is it? Ovarian locule or the ovarian cavity, right? Whatever you have inside, it is called as the ovarian locule or the ovarian cavity, right? Now inside this you will have something called as a placenta. What is this called? This is called as a placenta, and attached to this placenta you will have your ovule, right? Attached to this placenta you will have your ovule. Sometimes you will have just one ovule in an ovary, sometimes you will have more than one ovule in an ovary. It depends, right? So if you have just one ovary, right, have that understanding that ovary is the one which develops into fruit. Ovary is the one which develops into fruit, right? So sometimes you will have ovary is the one which will develop into fruit and ovule is the one which will develop into seed. So if you have one ovule, you are supposed to have one seed. If you have many ovule, you will have many seeds, right? So there are some plants which has many ovules in the ovary, and there are some plants which has only a single ovule in the ovary. Let's see some example. Now take this wheat, take mango, take paddy, right? Then what else? You can take papaya, then take watermelon, then also you can take orchid, right? How many seeds do you find in wheat, paddy and mango? You just find a single seed, which means they are the plants which has only a single ovule in the ovary. And look at papaya. You have many seeds. Watermelon also you have many seeds. Orchids also the same thing, right? So they are the ones which has many ovary, right? This way you can easily remember. See, examples is the place where they ask questions and it becomes very tricky at the end, right? Now let's move on to the Megasporangium. Megasporangium, understand, it is nothing but your ovule, this part. This is what you are going to learn, right? Yes, so let's move on to Megasporangium. Megasporangium is my ovule. Okay. Okay. Now, see, we had ovary, right? Inside that placenta, inside that ovule, right? Now, I am going to come from inside. Inside you have a structure like this, right? Around that you have one more structure like this. Then, so this is actually your embryo sac. What is this? This is your embryo sac. This is the one which is actually going to contain the gametes. And outside the embryo sac, I told you, whenever you have an important cell, you will have a nourishing cell around that. Just like sporogenous tissue and your tapetum, right? The same way here, embryo sac also has one more cell that is called as, just near to that, it is called as the nucellus. So what will be the function of nucellus? It will be for nourishment. It will have a multinucleated condition. It will help for nourishment and all, right? Fine. So I can write that also. It is for nourishment. Then outside that you have two protective coverings. One protective covering, right? One more protective covering. Yeah, this is how it looked like. Fine. So these two protective coverings are called as integuments. What are they called? So this will be your inner integuments, right? And outer to that you will have this one. This is your outer integument. Right? And what is the function of this integument? It is for protection. Protection of what? Even for sporogenous tissue, we had 3 layers of protection, right? The same way for embryo sac also we have protection, that is the outer and inner integuments, right? And you can find that there is a space here which is not enclosed by the integuments. So this particular space which is not enclosed by the integuments, it is called as your micropyle. What is it called? It is called as your micropyle, right? And this region, this end where you have the micropyle, it is called as your micropylar end, right? And the end opposite to the micropylar end, it is called as your chalazal end. Simple. Micropylar end and chalazal end. Yeah. Now this is ovule. So outside of that you are supposed to have the ovary. This is your ovary, right? And there is a stalk which attaches the ovule to the ovary. This stalk is what you call it as funicle, right? And what can you tell about funicle? It attaches the ovule to the ovary. Yeah, and this place of attachment, now if you see, this is the place of attachment of the funicle and the ovule. This place is what you call it as hilum. What do you call? This place is called as Hilum. So that is the structure of a megasporangium. You should know the structure, where is it present, and what is the function of that structure as well. Yes. So we started with embryo sac. Outside the embryo sac you have nucellus, the function is nourishment. Outside the nucellus you have two integuments, the function is protection. Yeah, and then there is a place in the ovule which is not enclosed by the integument, that is your micropyle. That end is called as your micropylar end. Opposite to micropylar end you have your chalazal end. The ovule and the ovary is connected by a stalk called as funicle, right, and the stalk is attaching to the ovule in this place called as hilum, right. So this is your megasporangium or ovule, right. Fine. Now let's move on to the megasporogenesis, nothing but the production of the female gamete. How is the female gamete produced? Yes, coming to my megasporogenesis. So what happens in megasporogenesis is that the normal cells of the ovule, what happens to that is it will differentiate, right? It will differentiate and it will form, or it will get matured into a megaspore mother cell, nothing but your MMC, right. And this happens in the micropylar region, very very important, right, in the micropylar region. Fine. After the formation of this megaspore mother cell, the megaspore mother cell will readily undergo meiosis, right. So if it undergoes meiosis, what will it form? It will form 4 megaspores, right. We know that there it was microspore, here it is megaspore, that's all. So as a result of meiosis you will end up having 4 cells, right. And let's see the ploidy also. This is the normal cells are 2n, the megaspore mother cell also is 2n. When it undergoes meiosis, definitely it is going to reduce the chromosome number, so this is n, right. So out of this 4 cells, right, you will have 4 cells, 1 2 3 4. 3 of that will degenerate and it will end up having only one functional megaspore. So what happens? It will have one functional megaspore, right, whereas the 3 will degenerate. Fine. So what happens to this one functional megaspore is that it will start undergoing free nuclear division. Now I want to make clear what is free nuclear division. See, when you take a cell division, first of all you will have karyokinesis, right. Karyokinesis is nothing but in a cell the nucleus, one nucleus would be there, that one nucleus will become 2 nucleus. That is division of nucleus is what you call it as karyokinesis. Then you will have cytokinesis. Cytokinesis is nothing but cytoplasm division of cytoplasm. So here what happens, the cytoplasm will divide and it will form 2 different cells, right. Free nuclear division means karyokinesis will happen but cytokinesis will not happen, right. So what is a free nuclear division? Free nuclear division is a division in which karyokinesis is not followed by cytokinesis. Simple as that, right. Now let's come back. So this one functional megaspore is going to proceed with free nuclear divisions, right, free nuclear divisions. So what happens? One cell will divide into 2, right. So how many nucleus will be there? 2 nucleate embryo will be formed. 2 nucleate embryo will be formed. Again this will undergo mitosis, right. See, this is also mitosis, because always remember mitosis will be present in, that is, see, meiosis. Meiosis can happen both in diploid cells and haploid cells. Sorry, meiosis can happen only in diploid cells, whereas mitosis can happen in both diploid and haploid cells, right. This also you have to remember, this small hint, not see, this is the way that you have to understand. No, not like that. This is a small hint wherein you can connect and learn. Yeah, so it will again undergo this mitosis. Again, this is also free nuclear, so it will form 4 nucleate embryo. Right, I'm continuing here. Again, this 4 nucleate embryo will undergo free nuclear division, and it will end up forming 8 nucleate embryo. Right, so you will have a single cell with 8 nucleus. That's your 8 nucleate embryo, and after forming this 8 nucleate embryo only, your cell wall formation will start. Cell wall formation. Right, so cell wall formation, what happens? The female gametophyte is formed after this, that is female gametophyte, or you can find that it is your matured embryo sac. Right, it is your matured embryo sac is formed. Right, now you have to know what are those cells. Right, so it looks something like this. This cell is your large central cell. Right, now I told you 8 nucleate embryo, right? So in that 2 nucleus are here, 1, 2, right? And you have 3 nucleus here, right? And you have 3 nucleus here. Now what are the cells? Here you have 3 cells aligned like this. These 3 cells are called as your antipodal cells. What is that? It is your antipodal cells, right? And these 2 cells, they are called as your, that's not a cell, that's just a nuclei. There is no cell wall around it, right? So this is your polar nuclei, right? Then you have these 2 cells. This is your synergids. What is that? Synergids. And this one cell, this is your egg cell. Fine, this is your egg cell. Now these 2 synergids and this egg cell together, it is called as egg apparatus. What is it? Egg apparatus. Right, then you have these 3 antipodal cells. So these 3 antipodal cells together, you call it as the antipodals. So this is the structure of a matured embryo sac, right? So how many cells does it has? It has 7 cells, right? And what are the 7 cells? 3 antipodals, 1, 2, 3, and 2 synergids, 1, 2, 1 egg cell, and the large central cell, right? So 7 cells, and how many nucleus? 1, 2, 3, 4, 5, 6, 7, 8, right? So it has 8 nucleus. So a mature embryo sac is 7 celled and 8 nucleate. Very, very important, right? So this is the structure of your embryo sac, and this is your megasporogenesis. Understand that all of these are free nuclear divisions, which means only karyokinesis, no cytokinesis, no cell wall formation, nothing, right? So this is your megasporogenesis. Yes, now let's move on to the diagrams that is given. So this picture is actually about your this is about your Megasporangium. Now if you can see this one, this is a not so developed one, right? So you have megaspore tetrad formed after meiosis, and in this 3 will degenerate, only 1 will be functional. That functional megaspore will develop like this. See, free nuclear division. This is your 2 nucleate embryo, this is your 4 nucleate embryo, this is, so this undergoes mitosis, right? This again undergoes mitosis. So you have 8 nucleate embryo. It starts to form the cell wall, and finally it looks like this. This is your final structure with the 7 cells and 8 nucleus. And this is another one picture. So this is of the gynoecium, or the carpel of a flower with stigma, style, ovary, and look at this thalamus, right? Thalamus is also there, and this is the ovary structure, and this is the structure of carpel in some plants, and this is the ovule before it gets matured. Yes. Now let's move on to some questions. So we'll discuss few questions. First question. In angiosperms, Megasporangium is equivalent to. So Megasporangium is nothing but the entire structure, right? So Megasporangium here is equivalent to, see, Megasporangium, actually you have anther. Inside anther you have Megasporangium. Inside Megasporangium you have sporogenous tissue, right? So it's like sporogenous tissue becomes pollen grains and Megasporangium becomes pollen sac. So it's the same thing here, ovule and ovary, right? Ovule you can relate it to pollen grain, and ovary you can relate it to the pollen sac. So in that case, Megasporangium here is nothing but the ovary. Ovary will be your Megasporangium. Next question. In angiosperms, embryo sac is direct question. You can easily answer. How many nucleus? 8 nucleus. How many cells? 7 cells. So it is 8 nucleate and 7 celled embryo sac when it comes to angiosperms. Next, choose the correct sequence representing the ploidy of nucellus, megaspore mother cell, megaspore, egg cell, zygote, polar nucleus of embryo sac, secondary nucleus, and primary endosperm nucleus. Okay, this question has from other topics also. So let's see. So you have these many options. So we know that zygote is definitely going to be diploid, right? And the primary endosperm nucleus, which is actually a result of fertilization. We'll learn it in post-fertilization structure, right? So this is 3N. This is triploid. So with this itself you can understand. And then secondary nucleus is going to be 2N, right? Then a polar nucleus is definitely going to be N. Then you have egg cell. Then you have egg cell. Egg cell is going to be, egg cell is going to be N. Megaspore is also N. Megaspore mother cell is 2N before meiosis. Then you have nucellus. Nucellus is also 2N, right? So with that your option will be option C. So you should know this. Yeah. Anyway, this primary endosperm nucleus alone, we'll do it in the next video. Yeah. Then X is a kind of stalk. It's a stalk that attaches an angiosperm ovule to placenta. So of course you have the ovule, you have the placenta. The one which attaches the ovule to placenta is funicle. Funicle, right? Funicle. Fine. Yeah, so X is funicle. We already know that. Body of the ovule fuses with X in the region called Y. Where is it fusing? It is fusing in this region called hilum, right? So what is X and Y? X is funicle, Y is hilum. Option A, direct question. But if you are not strong in your theory, you will get confused in even these simple questions. Yeah. Then your last question, function of filiform apparatus. Okay, we missed this part. Actually, you have the embryo sac and you have the synergids like this. You have the egg cell, right? So this one is your synergids, right? And what is this? This is your egg cell, right? And inside the synergids you will have a cellular thickening like this, right? What is it? It's just a cellular thickening, and this cellular thickening is your filiform apparatus, right? So the function of this filiform apparatus is that pollen tube will grow, right? During fertilization, pollen tube will grow and it has to reach this egg cell. So filiform apparatus will guide the pollen tube so that it goes and reaches the egg cell. So what's the function of filiform apparatus? Guide the entry of pollen tube. Very correct. Recognize the suitable pollen grain at stigma? No. Stimulate division of generative cell? No. Produce nectar? Not at all, right? So the function is to guide the entry of pollen tube to the egg cell, right? So with that, our topic, we are finishing this topic. So it is the female part that we have finished. We have completed the male part and the female part, and let's meet in the next video, double fertilization. Thank you.